Good afternoon, everyone. I'm Samson Eugin Simon from Liza Makowski Lab. I'm a postdoctoral fellow there. And I'm going to talk about exploring the impacts of methylation genetics on tumor suppressor in BXD preclinical mouse model. And breast cancer is a metabolic disease. It affects 1 in 8 women in USA. In that, TNBC is most aggressive, which accounts like 10 to 20% of all breast cancers. And TNBC is a heterogenic, and they are due to the aberrant risk genes or genetic modifiers. Genetic modifiers are a specific set of tumor suppressor genes or oncogenes that affects the phenotypic or genotypic traits in cancer. For an example, like RB1 or P53. And any abnormal epigenetic modifications will amplify oncogenes or downregulate tumor suppressor genes expressions, which results in cancer development. And in our lab, as everybody explained, BXD mouse model, so I got no words more to explain that. So we are just like crossing BXD mouse model between GEM, that is a spontaneous tumor mouse model which develops breast cancer, and we use BXD-BC model, that is cross between the BXD and the SV40 FBB3 C3 tag mouse model. Using that, we be collecting some phenotypic traits like survival, latency, tumor burden, multiplicity, and tumor volume. And I'm especially focusing on to the latency, which is the phase of initiation of tumor development. And using those data, we be interpreting them into a gene network to understand what QTL, and also we be doing some transcriptomic analysis on gene expressions along with this BXCBC strains. And based on the latency data or any other phenotypic traits, we found they are displaying significant heritability among BXT, different BXT strains. And on the right is the latency, in which is compared between the parent strain among different BXT strains, and found this tumor latency is variable among them, which are developing earlier to parent strain are considered to be more aggressive, and later are considered to be less aggressive. Interestingly, we found two different strains, that is 79 and 51, had never developed tumor. And we found that it's variable among 28 BXC strains. And based on the tumor latency, we did some gene network analysis and found chromosome 10 has the biggest hit. And based on the chromosome 10, we found around 7 set of genes. On that, RASF3 has a significant correlation towards latency. So looking deeper into RASF3, we looked into interaction partners using string and found they are interacting with their own family members. And also we found another gene that are a hit under chromosome 10 QTL analysis. In humans, it shows the same, and also it shows other interactions between oncogenes and tumor suppressor genes. And with this, we be narrowing down to a RASF family on further studies. RASF family is a tumor suppressor which exists in the cytosol of cancer cell. It's a scaffolding protein which interacts with RAS family or any other apoptotic family regulating the cell cycle, apoptosis, and cell division. This is least studied in breast cancer cells, so we are really interested to study in the breast cancers, like TNBC. And to understand the correlation between the RASA family and tumor latency, we did some correlation dot plot. and which is similar as a heat map too. And you can see the color represents the correlation between the tumor latency, and the size of the dots explains you the expression level of the genes. Uh, sorry. And based on the RASF3 identification from the QTL, like we also included a rest of the RASF family and correlated with latency. You can see from the left is the late latency and the right is the early latency, and which is positively correlating to most of the RASF family like RASF3, 6, 8, and 10. And also from chromosome 10 QTL hits, we found two other genes are like negatively correlated to the tumor latency. And to speak about DNA methylation in RASF family, we have this RASF1A that is well characterized in most of the cancer, which is like hypermethylated in most of the cancer. And the DNA hypermethylation happens in promoter region or initiator region, which turning off the transcription. And they are maintained by different methylation markers like writers, erasers, readers, or co-readers. Like significantly, we found RASF3 has some methylation region at the initiator site, which speculates us to know more on whether like RASF3 has a methylation in our variable expression between BXD strains. And we made a correlation plot between methylation markers and RASF family member genes in BXD strains. On the left is the overall correlation plot between latency and the multiplicity. And you could able to see the red and the green, that is like a positive correlation and the negative correlation. You could able to see some correlations between the methylation and the tumor phenotypes. And on the right, you can see a gene-to-gene correlation between the methylation markers and the RASF family like RASF family genes. And we found they are like positively and negatively correlating to each other, and which is like a lot more significant, which speculates as more for future studies to understand whether RASF family genes are like methylating. And interestingly, to understand whether RASF3 has a beneficial thing in TNBC human patients, we did a Kaplan-Meier plot analysis using KM plotter and found like RASF3 higher expressions saves like increases the survival of TNBC human patients, and also it benefits when it's treated with chemotherapy. And on the right, like you can also see RASF3 protein levels are like expressed variably among breast cancer cells. And for our conclusion, we developed a novel mouse model genetically programmed to develop BC, that is breast cancer, that displays significant variance in tumor phenotypes, and two unique strains, 51 and 79, that never developed tumor for 365 days. And based on the QTL identification, we did identify some candidate genes, especially RASF3. And also RASF3 is correlated with phenotypic traits in BXD-BC tumors, and RASF family is like correlated with methylation markers, which predicts possible methylation that attributes to tumor phenotypes. And yep, that's it. I would like to thank everybody, and also like to thank the organizers and my lab members, especially to Boston, Sandesh, and Jeremiah Hall for helping me throughout this study. And I also like to thank my PI, Liza Makowski. Thank you all.